Sushruta for NEET is now available on the Google Play Store. Try it out for free.
Hi, my dear students. Hearty welcome to JnanaMarga, your academy biology class. I am Prof G.K Bhat. Now let us continue the usage of microbes in human welfare. As we have seen in our last class, there are a good number of microbes which are quite useful to human beings. It is not that all microbes are harmful, and there are a good number of microbes which have made our life easy. Now let us look into the microbes which are used in the production of antibiotics. Antibiotics produced by microbes are regarded as one of the most significant discoveries of the 20th century and have greatly contributed towards the welfare of the human society. You may not be aware, lot of people used to die hundreds of years back, 100 years back, because of some of the diseases for which there is no treatment at all. You may wonder if I say, people used to die of malaria, people used to die of typhoid, people used to die of cholera. Forget about the smallpox. We don't have any such serious diseases now when compared to yesteryears. Now only the two diseases which are bothering very much, barring this corona or COVID-19, the other two diseases are, one is cancer and another is the AIDS. Here, antibiotics, what do you mean by that? Anti is a Greek word, it means against. Bio means life. That means against life. Against whose life? Not against we. It is against life in the context of disease-causing organism. These are the antibiotics are against the life of disease-causing organism. Whereas with separate human beings, they are pro-life. They save our life. They kill the disease-causing organisms, antibiotics. Antibiotics are commercial substances which are produced by some microbes, we shall see which microbe produces what antibiotic and can kill or retard the growth of other disease-causing microbes. Either they kill the disease-causing microbes or retard the growth of disease-causing microbes. You are all familiar with the commonly used antibiotic penicillin. Penicillin was the first antibiotic to be discovered. It is not a deliberate attempted discovery. Discovery of penicillin took place as a chance matter. Alexander Fleming, when he was working on Staphylococci bacteria, once observed a mold growing in one of his unwashed culture plate, around which the bacteria could not grow. What he was doing? Alexander Fleming had kept some of the vessels for the experiments he conducted. He was using some vessels and he kept some vessels unwashed, some plates. And what he could see, he observed a mold is here fungus, He observed a fungus or fungal member growing in one of his unwashed culture plate, around which the bacteria on which he was working could not grow. He was surprised why this bacteria Staphylococci did not go in that plate where some mold was there. He found that it was due to a chemical produced by the mold. This chemical produced by the mold kept this bacteria away, and he named it penicillin after the mold Penicillium notatum. What he observed? He observed the fungal member Penicillium notatum. This fungal member secreted some chemical. This chemical did not allow the bacteria to grow there. And he called this chemical produced by this Penicillium notatum as penicillin. Now, who will tell me to which particular group of Penicillium notatum belonged? Which fungal group? You know, class? Anyone? Anyone? Navya? Kanisa?
Yes, sir.
You know? Savitri?
Yes, sir.
Vismita?
Sir, is it phylum Ascomycete?
Phylum?
Sir, is
Who
it
is telling?
Ascomycete?
Who is telling?
Vismita.
Vismita, you are right, but not its phylum. It is a
Yes.
class.
Class.
Ascomyces. Ascomyces is correct. Do you know what all other classes under fungi?
Basidomycetes.
Hmm.
Phycomycetes.
Hmm.
Duetromycetes.
So, phycomycetes, ascomycetes, basidiomycetes, and deuteromycetes are the four classes. Yes, as you said, penicillium belongs to ascomycetes. See, this is what is required. You must remember all these classes, all these phyla, examples and all. Our time is running short. Don't waste your time. I have told you several times, please start writing the diary, bring to our notice what help you want. We are ready to help you. Each and every minute you should use very carefully, without wasting. This is what Staphylococci bacteria have. This bacteria did not grow where there was a penicillin. This is Penicillium notatum. However, its a full potential, that is, potentiality of penicillin, as an effective antibiotic was established much later by Ernst Chain and Howard Florey. Fleming was not knowing its medicinal value. It was only after Ernst Chain and Howard Florey showed that it is a very effective antibiotic. This antibiotic was extensively used to treat American soldiers wounded in the World War II. Very important, ask in the examination. In which war the penicillin was used extensively to treat the soldiers? In 1945, Fleming, Chain, and Florey were awarded the Nobel Prize for this discovery. And today, for each and everything, doctor will say, let us put antibiotics. Let us put antibiotics. So, in day-to-day life, we are extensively using these antibiotics nowadays. After penicillin, other antibiotics are purified from other microbes. Let us see which are these. Antibiotics have greatly improved our capacity to treat deadly diseases such as plague, whooping cough, calicancy, diphtheria, leprosy, which used to kill millions of people all over the globe. There was no medicine for plague, whooping cough, diphtheria, leprosy. Prior to this, antibiotics were discovered. Today, we cannot imagine a life without antibiotics. You just see, you know, some of you have studied. If you don't know, you can note down this. Plague is an infectious disease caused by bacteria called Yersinia pestis. Thousands and thousands of people used to die of a plague. Once the infection by plague started in a village, the whole village used to get killed by this bacteria. Pertussis, which is also known as whooping cough, is a respiratory disease, illness, very contagious disease caused by a type of bacteria called Bordetella pertussis. Bordetella pertussis. People used to die of whooping cough. Korean bacterium diphtheria is the bacterium that causes the disease diphtheria. Leprosy is caused by another bacteria, Mycobacterium leprae. So, in this way, you do come across many of these disease-causing organisms, and we have very effectively, we have the antibiotics for these bacteria. Please know all this, whatever I have given, important from the point of view of examination. Now let us look into chemicals, enzymes, and other bioactive molecules. Microbes helping in this. Microbes are also used for commercial and industrial production of certain chemicals like organic acids, alcohols, and enzymes. Examples of acid-producing microbes are Aspergillus niger. It is a fungus. Aspergillus belongs to, now it is not Vismita, it is someone else giving the answer. Why not boys? Hello? Anyone? Vishwas? Harshavardhan? Chinmayi? Prithvi? Rakesh? Siddesh? Charan, are you there? Have you come?
Yes, sir.
You have come?
Yes, sir.
Blessy? Blessy?
Yes, sir.
Are you there?
Yes, sir.
Kavita? Kavita? Kavita?
sir.
You are there?
Dance, sir.
Okay, don't
Yes,
come
sir.
late.
Sorry, sir.
Okay. Who will tell me? Aspergillus belongs to. Okay.
Ascomycetes.
Ascomycetes. Did you search? Did you search in the internet? Hello? So, you have this Aspergillus niger. It is known to produce citric acid. Always ask a question, which microbe produces what chemical? Acetobacter aceti. Easy to remember. Bacterium produces acetic acid. Clostridium botulicum produces butyric acid. And Lactobacillus produces lactic acid. All these are very important because lactic acid, butyric acid, acetic acid. Only one example that you remember is Aspergillus niger. It is not bacterium. It is a fungal member which is producing citric acid. Always you must remember this. This is what Aspergillus niger. As you said, yes, it belongs to the class Ascomycetes. Who told me the example? Who told that it belongs to Ascomycetes? Okay, Prithvi,
Hello.
can you tell me
Yes,
any
sir.
example for this? Ascomycetes, Namrata, Lohit,
Yes, sir.
Penicillium.
penicillin is okay. Apart from that, Neurospora. Apart from that,
Saccharomyces.
huh?
Saccharomyces.
Is it done in NCERT book? Apart from that, have you heard about the claviceps? Have you heard about the Neurospora? Yes or no?
Yes, sir.
Have you heard about another two?
Morels and truffles.
Yeah, these are all examples for Ascomycetes. Acetobacter aceti, known to produce acetic acid. Clostridium botulicum, known for butyric acid. Lactobacillus, lactic acid. Yeast, Saccharomyces cerevisiae, is used for commercial product of ethanol. Microbes are also used for production of enzymes. For example, lipases are used in detergent formulations. Whatever detergent you use, whatever the soap you use, they have lipases and are helpful in removing oily stains from the clothings. You must have noticed that bottled fruit juices bought from the market are clearer as compared to those made at home. Navya?
Yes, sir.
Have you noticed this? Bottled fruit juices bought from the market are clearer than those made at home.
Yes.
Are you aware?
Yes, sir.
Which one you have seen? Come on.
Fruit juices and all, sir.
Which fruit juice?
Real,
Huh?
like,
Pardon? Fruit juices bought from the market are clearer than what you make at home.
orange juices.
Orange juice, which bottle you will get?
So the brand,
Brand?
Real Tropicana.
Then they're all very
Fanta,
clear.
Mirinda.
Yeah?
Fanta and Mirinda.
Why you drink that?
I just told a brand name.
You don't use? Then how you can see that it is very clearer? Okay, fine. It is true. Whatever these bottle juices you see, they're always crystal clear than this. This is because the bottle juices are clarified by the use of pectinase and proteases. Important. Because of this, they are clarified by using these pectinases and proteases that make all these juices to be very clean. Remember this is important. Streptokinase produced by the bacterium Streptococcus, streptokinase, and modified by genetic engineering, let us see that later, is used as clot buster. Clot buster for removing the clots from the blood vessels of patients who have undergone myocardial infarction leading to heart attack. In these people who suffered from heart attack, the blood vessels which are blocked, this blockage or clot was removed by what is called streptokinase. It is commonly called, it is used as a clot buster. It used to break down this clot into small tiny particles and make the passage or flow of the blood easy. And what you should know, streptokinase is a clot buster and it is produced by the bacterium Streptococcus. Right? Okay? This is what you have. This is a normal coronary artery. You know, in myocardial infarction, it is not infection, myocardial infarction, the blood supply to the wall of the heart gets blocked. The blood vessels which are supplied to the wall of the heart are called coronary artery. The vessels which you take back are called coronary veins. To the wall of the heart, there is a separate blood supply. So whenever they get blocked, see here, this is a blockage you come across there by the deposition of cholesterol and all. And also you do see some of the block here. This is what is called atherosclerosis. Atherosclerosis is a blockage observed in the coronary artery. So these are cleared by making use of streptokinase produced by Streptococcus bacteria. Look here. This is what the clot you have. And this clot buster, it makes this clot into tiny particles which are always taken away in the flow of blood. Another bioactive molecule, cyclosporine. One more important, cyclosporine A, that is used as an immunosuppressive agent in organ transplant patient, is produced by the fungus Trichoderma polysporum. I hope you may be aware, whenever organ transplant is done, the recipient's immune system always rejects this new organ which is going to be administered to his body. So during kidney transplantation, most of the time during kidney transplantation is done between closest blood relatives. Even then, when an organ is transplanted into the body of the patient, the immune system of a patient's body, it tries to reject it. So in order to avoid it, in order to avoid this organ transplant rejection by the patient body, the patient is always administered what is called cyclosporine A. The moment cyclosporine A is injected or administered into the patient's body, his immune system gets suppressed. Once the immune system is suppressed, there is no question of rejection. I hope you are understanding my point. Is it clear? Is it clear, Jayasri?
No,
Let's see.
sir.
Huh?
No, sir.
No. Okay. Let me just reply. You know, generally, if at all we want to just transplant the kidney, now there is a patient who has a problem with his kidney, and doctors may say that he will not survive at all unless his kidney is replaced, or defective kidney is replaced with the normal kidney of the donor. This is what is called kidney transplantation. Many times, whenever a person suffers from kidney problem, generally the doctor try to see to treat him with one or the other medicine. Afterwards also, if at all it is not improving condition, then doctor may say someone has to donate the kidney. Who has to donate the kidney? Generally, donation is done between the closest blood relative, like daughter giving the kidney to, or sister giving the kidney to one of the other sister or brother, or father giving the kidney to the daughter, or vice versa. In the same, amongst the closest blood relatives, the kidney transplantation is done. But even then, when the kidney of the donor is put inside the body of the patient's immune system, thinking that it is the outside organ, it tries to attack it. It tries to reject it by attacking it. Immune system tries to attack it and it wants to reject it. It is only because of the immune system. Now what is done? Because this is going to happen, as we know very well, the patient is first administered with the immunosuppressant drug. When this drug is given, his immune system gets suppressed. His immune system will not work at all. It becomes, its hands are tied. So once the immune system is made weak, now the kidneys are transplanted, and this suppressed immune system, it cannot do anything now because it is already given some drug and it is suppressed. It is made helpless. So this drug is called Cyclosporine A, and this Cyclosporine A is produced by the fungus Trichoderma polysporum. Charan, understood? Is it clear, Janani? Hello?
Yes, sir.
Whenever we ask me or any other teacher, have you understood or is there any problem? At least one or two of you should immediately answer. Others will feel bad. Have you understood, Navya? Kanisa, Namrata,
Yes, sir.
Priti,
Yes, sir.
Priti,
Yes, sir.
Rakesh,
Yes, sir.
Sarvottam. Okay. So I hope you
sir.
have understood. Savitri, Artha Aitanamma.
Yes, sir.
Shall I go ahead?
Okay, sir.
So, this is the fungus trichoderma. But one thing you must remember, even though immune suppressant drug is administered, cyclosporine is administered, and kidney transplantation is done, because of the administration of this immunosuppressant drug, that patient, for each and everything, he should undergo for medication. He should immediately go to doctor and take relevant medicine for the problem. Minor problem. If at all he suffers from a cold, he has to go to doctor. If at all he suffers from headache, he has to go to doctor. If at all he suffers from fever, he has to go to doctor. For each and every ailment, he has to go to doctor because his immune system is already made defunct. Okay. Coming to this is the fungus trichoderma. Always ask a question. Cyclosporine is produced by the fungus, or in one column cyclosporine is given, and in another column this what? Immune suppressant drug may be mentioned. Like that, whatever you have studied, all the different types of bacteria name will be given at one side. The whatever the drug they are giving, that will be made, or to which they are concerned, that may be informed or given. So these are the things. You have now what is called statins. Statins. Statins produced by the yeast Monascus purpureus have been commercialized as a blood cholesterol lowering agent. So statin is administered to the patient to lower the blood cholesterol level. So this statin, though sometimes many times doctors say cholesterol level is high, cholesterol level is high. Such patients who have high blood cholesterol level are given the statins, and these statins are produced by Monascus purpureus. It is a yeast fungal member. You must know whatever we are looking at, whether it's the fungal member, whether it's a bacterial member, you must know. It acts competitively inhibiting the enzyme responsible for synthesis of cholesterol. Okay? It acts by competitively inhibiting the enzyme responsible for the synthesis of cholesterol. So cholesterol level comes down. Right? Statins are a class of drugs often prescribed by doctors to help lower cholesterol level in the blood. By lowering the levels, they help prevent heart attack and stroke. You know, if at all anyone's cholesterol level goes up, it may result in heart attack and stroke. Studies show that in certain people, statins reduce the risk of heart attack, stroke, and even death from heart disease by about 25 to 30%. Now let us look into microbes in a sewage treatment. Right? You know, disposal of sewage is the greatest burden in almost every city because of the population explosion. We know that large quantities of wastewater are generated every day in cities and towns. This wastewater also contains human excreta. The major component of this wastewater is human excreta. Excreta is a fecal matter. Urine. This municipal wastewater is also called sewage. It contains a large amount of organic matter and microbes. And many of these microbes are pathogenic disease causes. Now, let us see how to treat that. Have you ever wondered where this huge quantity of sewage or urban wastewater is disposed of daily? No, we never bother because we don't have any time to know about all that things. But it is sure that from every city, it should get disposed of daily. This cannot be discharged into natural water bodies. Sewage cannot be discharged into natural water bodies like rivers and streams directly because they cause death of these microorganisms and also the individuals there. Health problems and diseases have often been caused by discharging untreated or inadequately treated sewage to the natural water bodies. Not only that create problem to the aquatic organisms, even they bring about health problems and diseases. Before disposal, hence sewage is treated in sewage treatment plants, what is called STPs. Sewage treatment plants. What is this sewage treatment plant? How is this sewage treatment plant? We have to look into that. So before the disposal, in every city, sewage is treated in sewage treatment plant, what is called STPs, to make it less polluting. I don't know whether you are aware or not. In all big cities, the apartments, each and every apartment with more than 100 or 200 houses are required to maintain their own STPs. Every apartment has their own sewage treatment plant. In my apartment, we also have a sewage treatment plant. Now let us see how this sewage treatment plant works. Treatment of wastewater is done by the heterotrophic microbes. Microbes which are dependent on others for the food, which do not synthesize their food on their own. Treatment of wastewater is done by the heterotrophic microbes naturally present in the sewage. Microbes only, they treat the sewage. Treatment of wastewater is done by the heterotrophic microbe. Autotrophic microbes are the one which can synthesize their food on their own. Heterotrophic microbes are the one which are dependent on others for the food. So this treatment of wastewater is done by heterotrophic microbes. We shall see which are they, and how they work. This treatment is carried out in 2 stages. Sewage treatment in STPs is carried in 2 stages. Let us see what are that. Primary treatment. These treatment steps basically involve physical removal of particles. Primary treatment involves physical removal of particles, large and small, from the sewage through filtration and sedimentation. What is done? The sewage water is made to flow. When it is flowing for some distance, solid particles start getting settled at the bottom, and the light particles and all, they start floating, and these all floating ones are removed by filtration or through filtration, and the solid ones are made to undergo sedimentation. So this is the primary treatment. The primary treatment involves steps basically which are concerned with the physical removal of particles, maybe of large and small size, and remove particles from sewage through filtration and sedimentation. Physical removal of particles, large and small, from sewage through filtration and sedimentation. These are removed in 2 stages. Initially, floating debris is removed by sequential filtration, one after another, making it to undergo filtration several times. So by this sequential filtration, floating debris are removed. Floating debris is removed. Okay, that is the primary treatment. Then the grit, that is the whatever that you come across in that soil and the small pebbles, are removed by sedimentation. So all the lighter particles, floating particles, are removed by filtering, sequential filtration, and the small soil and other small pebbles are removed by sedimentation. All solids that settle form primary sludge. So many of the solids, they settle at the bottom. And these solids which settle at the bottom forms what is called primary sludge, and the supernatant forms effluent. Supernatant forms effluent. Suppose we make it some water, sewage water to flow. Okay, you see that all small soil, small pebbles and all, they collect. They collect at the bottom. And clean water, they collect bottom. Clean water, supernatant water, flows on the upper side. And this, whatever that is flowing as a supernatant form, is called effluent, and down below is a primary sludge. The effluent from the primary settling tank, effluent, that is this one. Effluent from the primary settling tank is now taken to secondary treatment. Secondary treatment. Okay. Rakesh,
Yes, sir.
tell me what is done while a treatment is given in the STPs.
Sedimentation, and then filtration.
Yeah, sedimentation and
Filtration.
what is done? What is
In
happening?
sedimentation, large particles are settled down when the sewage water is flowing, and then in the filtration, the small particles are filtered.
Small particles means what? Small particles are filtered. Which are removed by sequential filtration. By sequential filtration, floating debris are removed. Okay? Now, this is what you have. Look into the diagram. This particular part concerned with the, what is its primary? What is the primary? What is the secondary? Namrata?
Treatment.
Treatment. In the primary treatment, what you get at the bottom? Sediments.
Sediment.
And on the top, you come across floating debris. They are made to pass from one tank to tank by sequential filtration. And what you call this? What is this and what is this? This is effluent. And this is
Primary sludge.
primary sludge. Okay. Now this effluent is made to go to secondary treatment. Okay. So the effluent from the primary settling tank is taken to the taken for secondary treatment. Okay. Now, secondary treatment is otherwise called biological treatment. What is happening now? The primary effluent is passed into large aeration tank. Large aeration tank, which one? Primary effluent is passed into large aeration tank where it is constantly agitated mechanically and air is pumped into it. Very, very important. So this primary effluent, a clear supernatant, is passed into a large aeration tank where the water is, effluent is constantly agitated, shaken, and also air is pumped into it. This allows, very important, is agitated constantly, mechanically, by connecting it to the power source, and air is pumped into it. This allows vigorous growth of useful aerobic microbes into flocks. This constant agitation, mechanical agitation, and the pumping of air into it allows vigorous growth of useful aerobic microbes into flocks. What is this flocks? Masses of bacteria associated with the fungal filament. So form mesh-like structure. So whenever there is a whenever there is a constant agitation and mechanical agitation and air is pumped, you do come across vigorous growth of bacteria and fungal filaments forming what is called flock, mesh-like structure. While growing, these microbes, bacteria and fungi, consume major part of the organic matter in the effluent. Major part of the organic matter in effluent. This significantly reduces BOD of the effluent. What is that BOD? Biochemical oxygen demand. The moment microbes use more and more of the organic matter, of the sewage, or the effluent, the BOD, biochemical oxygen demand, it comes down significantly. Right? Is it clear? So what we have done? We have made the primary effluent to move from the primary treatment to secondary treatment plant. After bringing them here, this particular primary effluent is constantly mechanically agitated and air is pumped into that. This encourages growth of more and more of these microbes, and these microbes consume a major part of organic matter in the effluent, and thereby the BOD, biochemical oxygen demand, comes down. So this is what is aeration tank. What is this? What is this? What is coming through this? What is coming through this?
Primary effluent.
Primary effluent. It has come from where? It has come from primary treatment tank to here. Now, what is this? What is this arrow mark indicates? Arrow mark indicates there is constant mechanical agitation, shaking, constant mechanical agitation. And what is done at the same time? Air is pumped. And both this brings about what? It makes the microbes appear, make use of all this organic matter, and thereby, if you look into this, either earlier the sewage had high BOD, biochemical oxygen demand. Now the biochemical oxygen demand level comes down. This means lesser the BOD in any water, more pure it is. Higher the BOD in any water, it is more impure or polluted. So now it is made to pass out. Okay. So this is secondary treatment plant. You can see here, you come across agitation, shaking. You can see some sort of ripples appearing there in the water. Okay, right. And you do come across a flux there. It is the mesh-like structure found by bacteria and fungi. BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of 1 liter of water were oxidized by bacteria. So, BOD refers to amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. Whenever sewage is discharged into any part, then the BOD will be very high. Biological oxygen demand or biochemical oxygen demand will be very high. Whenever the water gets purified or sewage gets purified because of the microbes and all, by using the organic matter, the oxygen demand comes down, BOD level comes down. The sewage water is treated till the BOD is reduced. The BOD test measures the rate of uptake of oxygen by microorganism in a sample of water, and thus indirectly BOD is a measure of organic matter present in water. High BOD means more organic matter. Low BOD means less organic matter. The greater the BOD of wastewater, more is its polluting potential. Are you understanding this point? Have you understood? Ditya?
Yes, sir.
Okay. Chinmayi?
Yes, sir.
Aisha? Aisha,
Yes, sir.
are you there?
Yes, sir.
Okay. Shall I go ahead? Savitri?
Yes, sir.
Shall I go ahead?
Yes, sir.
Okay. Once the BOD, hello? No, sorry. Okay. Hmm. 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 ನಾಲ್ಕರಿಂದ 5:45 ಸರಿ 9 yeah okay sir okay so coming to this once the BOD of sewage or wastewater is reduced significantly, the effluent is then passed into settling tank where the bacteria flocks are allowed to sediment to get settled. So after passing to second treatment, the sewage or wastewater is reduced, BOD is that wastewater reducer, and effluent is then passed into settling tank, where bacterial flocks are allowed to sediment, and this sediment is called activated sludge. So a part of this activated sludge, small part of the activated sludge, is pumped back into the aeration tank to serve as inoculum. What is this aeration tank? Siddesh? Siddesh, what is this aeration tank? What is happening in the aeration tank? Siddesh?
Sir, the influence is
Pardon? Aeration tank ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ? Harshvardhan
Yes, sir.
or Vishwas? ಏನು ಹರ್ಷವರ್ಧನ್? ಏರೇಷನ್ ಟ್ಯಾಂಕ್ ಅಲ್ಲಿ ಏನು? ಏರೇಷನ್ ಟ್ಯಾಂಕ್ ಅಂದ್ರೆ ಏನು? What comes to aeration tank? What is happening in aeration tank? Who will answer this question?
Sir, the primary effluent are constantly mixed and air is pumped on it.
Primary effluent comes to the aeration tank where primary effluent is mechanically
Mixed or
mechanically agitated and air is pumped into that. So that what will happen?
Microbes will grow in it.
Microbes will grow. So what we do to enhance the growth of microbes from the activated sludge, small part of the activated sludge is just added to aeration tank. Why that small part of the activated sludge is added to the aeration tank? Kavita? Nadira?
Yes, sir.
Yeah. Why is that? Who is this? Kavita or Nadira?
Kavita, sir.
Kavita. Tell me, why that small part of activated sludge is added to aeration tank?
To act as inoculum.
Huh? Yeah, what is there? What is there in that activated sludge? What is there in the activated sludge?
Bacterial flocks and
Bacterial flocks are there. So just to see that in the aeration tank, the bacterial that flock containing bacteria and fungal are growing quickly. We are using this certain part of bacterial sludge to aeration tank and they serve as inoculum. The remaining major part of the sludge, activated sludge, is pumped into large tanks called anaerobic sludge digester. So little bit is added, used as inoculum, but a large amount of activated sludge, they are pumped into a large tank called anaerobic sludge digesters. What will happen here in the anaerobic sludge digesters? Here, in aerobic sludge digesters, other kinds of bacteria, methanogenic bacteria, which grow anaerobically, digest the bacteria and fungi in the sludge. Whatever bacteria and fungi is there, they are getting digested by another kinds of bacteria. Methanogenic bacteria, methane producing bacteria. During this digestion, bacteria produce a mixture of gases such as methane, hydrogen sulfide, and carbon dioxide, and these form the gases of biogas. These gases form biogas and can be used as a source of energy as it is inflammable. Right? So this is what I said. So now primary treatment, the primary effluent is coming to the aeration tank. Here it is mechanically continuously agitated and air is pumped. Then it comes here, and when it comes down here, it is made to flow here. And now you have activated sludge. Certain part of activated sludge is taken back and put to this aeration tank. And others come down, and this is what is known as the what is that? What is it called? Sludge. What is that? Anaerobic sludge digester. In this anaerobic sludge digester, much of the bacteria and fungi are digested by methane bacteria. Okay. At that time, gases they are released, methane, hydrogen sulfide, carbon dioxide, which are inflammable, used as biogas. Understood? The effluent from the secondary treatment. Now, whatever is going now from here, secondary treatment, the effluent from secondary treatment water, this is actually pure water. This water, it can be used for agriculture purpose. The effluent from the second treatment plant is generally released into natural water bodies like rivers and streams, okay, and can be used for agriculture purpose. And this is what you have, the aerial view of the sewage plant, and this is what you see in big cities, sewage treatment plants. Okay, is it understood? Janani,
Yes, sir.
clear? Navya, Namrata,
Yes, sir.
Lohit, Lohit,
Yes, sir.
shall you go ahead?
Yes, sir.
Anyone wants any clarification anywhere? No. You can appreciate how microbes play a major role in treating millions of gallons of wastewater every day across the globe. This methodology has been practiced for more than a century now in almost all parts of the world. Till date, no man-made technology has been able to rival the microbial treatment of sewage. No. Only we are always dependent on this microbial treatment of the sewage. You are aware that due to increasing urbanization, conversion of more and more villages to city, township, sewage is being produced in much larger quantities than ever before. However, the number of sewage treatment plants has not increased enough to treat such large quantities. So, the untreated sewage is often discharged directly into the rivers, leading to their pollution and increase in waterborne diseases. So here and there, we always come across waterborne diseases because of mixing of the sewage water with the potable or drinking water. The Ministry of Environment and Forests has initiated Ganga Action Plan and Yamuna Action Plan, important two action plans, to save these major rivers of our country from pollution. Save Ganga, to save Ganga, Ganga Action Plan. To save Yamuna, Yamuna Action Plan. To save these major rivers of our country from pollution. Under these plans, it is proposed to build a large number of sewage treatment plants so that only treated sewage may be discharged in the rivers. A visit to sewage treatment plant situated in any place near you would be very interesting and educating experience, and you can do that after writing the NEET exam. Right? Now let us look into microbes in production of biogas. Biogas is a mixture of gases containing predominantly methane, produced by the microbial activity and which may be used as a fuel. We are using biogas, okay, gobar gas. We are using gas cylinder that is also inflammable. Biogas is the one which is produced by the living organism, biogas. You have learned that microbes produce different types of gaseous end products during growth and metabolism. The type of the gas produced depends upon the microbes and the organic substrate they utilize. I hope you have understood. The type of gas produced depends upon the microbes and the organic substrate they utilize. Of course, this you might have felt in your day-to-day experience. Am I right? You eat a large quantity of this potato. Any potato you do, sweet potato or potato. Okay? Okay. Can you get to know very next day or after some time? Hello? Are you understanding my point? If you eat more of potato, more of sweet potato, you start producing gases. Okay? More gas starts coming from you. Okay? So whatever organic substrate you utilize, you come across accordingly different gases. In the example cited in relation to fermentation for dough, cheese making, and production beverages, the main gas produced was CO2. Yesterday you have seen, in the batter, you come across puffy appearance and holes and all. What gases comes? CO2. However, certain bacteria which grow anaerobically on cellulosic material, you eat more broccoli. Have you heard about broccoli? Have anyone of you heard about broccoli?
Yes,
Yeah?
sir.
You do one thing. you eat extensively broccoli, and you see who will sit nearby you next day. Okay? Which go anaerobically on cellular material produce large amount of methane along with the CO2 and H2. Okay? Methane means you can't see it near that person. Okay? So cellulose material, if the more and more cellulose material is produced, more large amount of methane, CO2 and H2 gets produced. Okay? So Methanol bacterium. These bacteria, methanogenic bacteria, are collectively called methanogens, and one such common bacterium is a methanobacterium. What methanobacterium does? It produces a methane. Collectively called methanogen. Gen is production. These bacteria are commonly found in anaerobic sludge during sludge sewage treatment. Anaerobic sludge during sewage treatment. Anaerobic, you know that. Okay. Now, these bacteria are also present in the rumen, a part of the stomach of the cattle. You know, cattle has a four-chambered stomach. This is the first chamber, rumen. This is the second chamber, reticulum. Third chamber, omasum. And fourth chamber, abomasum. Right? So, in the rumen here, in case of cattle, you would come across the bacteria, methanogen bacteria to be present. A lot of cellulose material present in the food of cattle is also present in the rumen. So here, actually, in the rumen part, the methane gets produced. That is why the cow dung smells methane. In rumen, these bacteria help in the breakdown of cellulose and play an important role in the nutrition of cattle. So cow dung has its own smell, and it is because of this methanogen bacteria there releasing methane. Do you think we human beings are able to digest the cellulose present in our food? Can you digest? Hello, yes or no?
No, sir.
No. Yeah, you do one thing. You eat cellulose-rich food. Your basale, harive, something. Cellulose-rich in food. Next day, if at all you look into feces, what will happen? Which color appears? Which color appears? Which color appears? Hello? No one is ready to tell. I will not ask your name. Which color appears? You don't know? You do tomorrow. You just eat more of this leafy vegetable and see what will be there in next day. You know very well, it will be greenish in color. Thus, the excreta, dung of cattle, commonly called gobar, is rich in this bacteria. Dung can be used for generation of biogas, commonly called gobar gas. Dung is cow dung. Okay? See this for some time. Read this. Can you see this properly? Are you seeing?
Yes, sir.
Over. Foods high in fiber, such as broccoli, may cause a flatulence to be smelly. She is resting coolly. Poor guy. Husband and wife watching TV. No courage to walk away. He'll be blasted. See that? May cause flatulence to be smelly. The fermentation process produces odorous gas. In some cases, foods higher in the fiber have a distinctive odor. The natural odor from these foods can also cause flatulence to be smelly. You eat a little bit more of garlic, you'll experience that. Okay, right? So this is the composition of your flatulence. How much is there? Different gases. Okay, if you want, you can have the screenshot and watch it. Look into that slowly later. Shall you go ahead?
Yes,
Okay. So average person will fart 14 times a day. I hope you know what is farting. Okay, all right. Shall I go ahead? Okay, come. The biogas plant consists of concrete tank. This is what biogas plant will be asked sometimes for 4 5 marks question. Biogas plant, biogas plant consists of concrete tank 10 to 15 feet deep. Okay, in which biowaste are collected and a slurry of dung is fed. Here you see that dung with water is mixed and put here. A floating cover, this is the floating cover. This is what your floating cover. A floating cover is placed over the slurry, which keeps on raising as gas is produced in tank due to microbial activity. This will be tight. So this is the floating uh, cover, and this is what you have. This concrete tank. There will be provision to let this slurry to enter here inside. The biogas plant has an outlet. This outlet which is connected to a pipe to supply biogas to the near a house to the kitchen like that for either kitchen that cooking purpose or for lighting purpose. Okay, I hope it is understood clear. So the gases listed are this what methane and that of carbon dioxide. Okay, so the spent slurry is removed. This is what the spent slurry. It comes here, removed through another outlet and may be used as fertilizer. So when the gas gets accumulated, this will come up and this gas is carried to the kitchen for fire, cooking or lighting purpose. Cattle dung is available in large quantities in rural areas where cattle are used for a variety of purposes. So biogas plants are more often built in rural areas. The biogas thus produced is used for cooking and lighting. The picture biogas plant is shown here. I hope you understood this. Very important for labeling and also for board examination question. With a neat label diagram, describe this biogas plant. Right? The same thing. Now, the technology of biogas production was developed in India mainly due to efforts of Indian Agriculture Research Institute, IARI, and Khadi and Village Industries Commission, KVIC. Important to remember. If your school is situated in a village, you just make it a point to see how it works. Right? Now, microbes as biocontrol agent. Biocontrol refers to the use of biological method for controlling plant diseases and pests. We control the disease pests by using biological weapon. In modern society, these problems have been tackled increasingly by the use of chemicals, by use of insecticides and pesticides. So generally, we are going for use of chemical. If at all we want to control the diseases and pests. Pests are nothing but unwanted guests. Now, what we do, we just spray some chemicals like insecticide, pesticide, herbicide, etc. But it is not, it is only treatment to the chemical, but we don't want. We want microbes to treat these disease-causing organisms or disease and pests. These chemicals are toxic. Whatever chemicals we use, they are toxic. Herbicide, pesticide, insecticide, etc. Extremely harmful to human beings and animals alike and have been polluting our environment, soil, groundwater, fruits, vegetables, crop, plants, etc. So if at all you spray some chemical on one particular crop, this chemical spreads to other areas. So whatever it brings about pollution of soil, groundwater, etc., and also it brings about the pollution of our eatables. Our soil is also polluted through our use of weedicide, which we are using to remove weeds. Weed is unwanted plants. Biological control of pests and disease. Let us see how that biological control is done. In agriculture, there is a method of controlling pests that relies on natural predation rather than introduced chemical. Without introducing chemical, without putting the chemical, the pests can be or the disease-causing organisms can be controlled by biological method. A key belief of the organic farm is that biodiversity always furthers health, it improves health. The more variety a landscape has, more sustainability is. Always having luxuriant biodiversity is good for us. The organic farmer, therefore, works to create a system where the insects that are sometimes called pests insects, which they are creating some harm to us, The insects that are sometimes called pests are not eradicated completely, but instead are kept at a manageable level by a complex system of checks and balances within a living and vibrant ecosystem. Now, what does it mean? If at all you see that a particular insect, say A, is harming our crop, some crop it is harming, yeah, that we don't want to completely eradicate this A pest which is harming your crop, pest, insect is harming. We don't want to completely remove it. Rather than we want to control its population. We wanted to keep it under control rather than completely removing because this insect may be food for another insect or any bird. It may be serving as a food. If at all we remove it completely, the bird will not get the food. Another problem arises. So it is a check and balance. We don't eradicate completely. On the other hand, a check and balance is maintained. Contrary to the conventional farming practice means the good old method which we used to do. Contrary to the conventional farming practices, which often use chemical methods to kill both useful and harmful life forms, whenever chemicals are applied, not that only harmful organisms will be killed, even useful organisms may be also killed. So, contrary to conventional farming practices, which often use chemical methods to kill both useful and harmful life forms indiscriminately, this is a holistic approach that seeks to develop an understanding of the webs of interaction between the myriads of organisms that constitute the field fauna and flora. So, whenever we are using some chemical to kill one unwanted pest, at the same time that while killing that, some other which are not harmful to us, they may also get killed. So we don't want to kill other organisms because they have their own role to play in the environment. So this belief makes us to go for some other method. So we are not using the chemicals to kill this pest, but we are using the biological method to control what we don't want or our pest. The organic farmer holds the view that the eradication of creatures that are often described as pests is not only possible, that often described as pests is not only possible but also undesirable. Not only possible but also undesirable. For without them, the beneficial predatory and parasitic insects which depend upon them as food or foods would not be able to survive. Though that's what I said. Whenever we are killing this A, that B is dependent on that. So this will be trouble. So we don't want to go for that, right? Thus, the use of biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. What is this use of a biocontrol measure? An important part of the biological farming approach is to become familiar with the various life forms that inhabit the field, predators as well as pests, and also their life cycle, patterns of feeding, and the habitats that they prefer. So if at all you want to adopt some biocontrol measure, you should know which particular organism eaten by which is its predator, which is its prey, what is the life cycle, what is the pattern of feeding of that particular organism, where it occurs. All these you should know if at all you are going to develop one biocontrol measure. This will help develop appropriate means of biocontrol. Okay, now let us see. The very familiar beetle with the red disease, beetle with the red and black color, is useful to get rid of aphids. Oh, yesterday I said to see the aphids. Have you seen any one of you? Canisa? Canisa?
Yes, sir.
Have you seen the aphids?
No, sir.
For God. Anyone? Anyone try to see aphids? Anyone? Okay, right. So, very familiar beetle, red beetle, is used to get rid of aphids. So, you need not to spray anything on this to kill the aphids. You leave some beetle there, beetle will eat the aphids. Okay, because only these aphids are harmful to us, to one or the other vegetable or one or the flower. Not this beetle. Beetle is not going to create any problem to us. So, you may say that then beetle population increases. This beetle may be eaten by another bird. So we need not bother much about it or some other organism. So the dragonflies are useful to get rid of mosquitoes. Dragonfly kills mosquitoes. So this is what is called biocontrol. Have you understood? An example of microbial biocontrol agents that can be introduced in order to control butterfly caterpillar is the bacteria Bacillus thuringiensis. So many times, caterpillar larvae, they always harm our vegetables or fruity trees, etc. Caterpillars always feed on leaves, and adults feed on nectar. Okay, I shall just stop it here, and we have to take a little bit another 10-15 minutes, and in the next class I am going to take up the biotechnology chapter, and I'll be putting this in our Google Classroom. Please start answering to that, and we'll be taking up in our next week classes MCQs discussion on strategies of the food production strategies for enhancement in production, and this also. For your coming test on Sunday, this chapter is not there. Chapter 1 to chapter 9. Okay? Okay?
Okay, sir.
Okay, all the best. I think I will not meet you prior to your test. All the best. It is CET test. Do well. Okay. Any doubt you have? Anything you want to ask me?